Thanks for the invitation to speak. I want to thank Thermo Fisher, and I'll thank them again for early access to this new platform. So I'm David Berman, as was mentioned before, and I come from lovely Queen's University on Lake Ontario in Canada. Disclaimers, Thermo Fisher doesn't guarantee that anything I say is true, and they provided travel support but no remuneration. So today I'm going to talk about our work in prostate cancer biomarkers, diagnostic biomarkers, mainly focused on who we should biopsy, and prognostic biomarkers to help stratify treatment. Currently, prostate cancer is diagnosed primarily on the basis of the PSA test. So rising prostate-specific antigen levels in the serum of men triggers a need to biopsy. Digital rectal exam is also used, but it's a lot less accurate. So PSA is very sensitive, but not very specific. Tissue diagnosis is needed, as is the case for all solid tumors. Just in relation to the last talk, I want to mention that these are tiny localized tumors, about maybe a gram in total, as opposed to maybe 100 or 1,000 times more tumor burden in the metastatic patients discussed in the last talk. So this barbaric procedure involves inserting a 2-centimeter needle through the rectum into the prostate 10 or 12 times in the patient. And as you can imagine, there are serious complications. In about 1% of men, they will end up in the hospital. There's even an increased risk of death in some patients. Now, this 1% doesn't sound like a lot, but there are up to 1 million biopsies per year in North America. So we're talking about maybe 10,000 men every year undergoing serious complications. So these thread-like biopsies seen here are about 1 millimeter in diameter, and they're looked at under the microscope by pathologists. My postdoc provided this image. I think he really nailed it. This is what pathologists like me look like. And we look under the microscope and we decide, is there cancer? And if there's cancer, is the grade high or low? So before we get into methylation, I just wanted to mention that if you think about prostate cancer biomarkers, we don't have recurrent mutations in many cases, and the genomic aberrations in these early cancers are not very rich. And the one exception is methylation. DNA methylation at CpG islands is ubiquitous. There are hundreds or thousands of such events, very well characterized, very stereotyped in prostate cancer, and we wanted to see what we could do with those. Now, there are some challenges to working with methylation. Most methods for detecting methylation rely on bisulfite conversion, which converts unmethylated cytosines to uracil, whereas the methylated cytosines are protected. So the resulting changes in the DNA sequence can be recognized by hybridization or sequencing. The problem is it's a very harsh treatment. It lowers your yield quite a lot, and what you do get is degraded. So you need a very robust system for detecting it. Traditionally, we've used methylation-specific PCR, which has downsides that I'll talk about later. In sequencing, you need a reference genome to compare to, and that's not been a readily available resource. Thermo Fisher has kind of solved that problem. So using methylation-specific PCR, we did a big study just to ask the question, how accurately can you detect prostate cancer using methylation? We did this in tissue, which is the gold standard, so that we knew what's the maximum possible performance. This study was led by a graduate student, Palak Patel. This was part of his PhD thesis. He's since left the lab, and we're looking for a replacement, a student or a postdoc. If you know anybody who you think would like to get involved in work like this, please send them our way. That'll be my remuneration. So this was published earlier this summer, and we profiled 14 different loci and looked at all possible combinations. We got an AUC curve you don't usually see, above 90% accuracy. We had separate training and validation cohorts. The independent validation cohort was not used to train the classifier, and in that independent validation cohort, we got 94% sensitivity and 93% specificity. All told, there were 699 prostatectomy specimens in this study. So what can we do with that information? Well, pathologists like me can actually tell you quite accurately by looking at a slide like this, is cancer present or not present? So it's not that useful to have a tissue classifier, but what it gives us is a candidate liquid biopsy biomarker, and so that's a goal, is to translate that into urine as our non-invasive biopsy, and we'll talk a little bit more about that later. The other piece of information we can get, or I can get out of this image or out of a microscope slide with prostate cancer, is the grade. So low-grade cancers, those cells arrange themselves in discrete circles like these. And every cell in this circle has pretty much an equivalent environment and behavior, and that behavior is very indolent and harmless. Whereas high-grade cancers have these, instead of these discrete glands, they're fused, or the cancer cells don't form glands at all. So the ability to sort of break these rules and grow in all these different patterns heralds the ability to grow outside of the prostate as a metastatic nodule, and the ability to kill patients. So you might notice, well, a couple of things that I'd like you to notice. One of them is that it seems pretty clear that these are well-formed circles and these aren't, but what about these and these? It turns out that prostate cancer grading can be very subjective, and even the most expert pathologists can disagree with each other. The other thing I'd like you to notice is that depending on where you took this biopsy, let's say you took it in this plane or in this plane, you might get a very different ascertainment of what the grade was. And so that's something called sampling error, which is another big problem with biopsies, and one we thought we could address along with the subjectivity using biomarkers. So why does that matter? It turns out that if you look at it turns out that Gleason grade from prostatectomy, entirely removed prostates, is the most powerful prognostic biomarker for prostate cancer. So if you have, mostly high-grade cancers, high-grade cancer cells, that is, your risk of death is shown by this black area 20 years after your prostate comes out. And so most men will die of prostate cancer, shown in black, as opposed to other causes shown in gray. If you have no high-grade elements, you're pure low-grade, black is nearly invisible. Almost everybody dies of something else. And therefore, those men are offered something called active surveillance, where they're monitored instead of treated. That's the gold standard now in North America. So active surveillance involves regular digital rectal exams, PSA testing, biopsies, sometimes imaging. And the problem with this is about half of men will fall off of active surveillance within 3 years because they're recognized as having a higher grade cancer that was undetected at the beginning of their active surveillance. And that's been shown, or it's at least believed by most people, not to be progression of the cancer, but just a problem in detection or diagnosis. Again, that's a role for a biomarker, and I'm lucky to co-lead a pan-Canadian team project to address this problem, creating a Gleason grade biomarker that distinguishes between men with pure low-grade cancer and men with higher grade cancers. So we used a candidate gene panel approach. None of this was Thermo Fisher. We measured transcripts from pathways that are well established as being linked to prostate cancer aggressivity. We combined them with copy number and DNA methylation features. So this is a new approach where we're trying to combine RNA and DNA, and we know that commercial panels now can do that kind of thing, although not yet clinically for methylation, or at least not at the time. To address sampling error, we sampled every prostatectomy twice in 2 different areas and profiled them separately. When there were high-grade components and low-grade components, we enriched one for low and one for high. We again had separate training and validation cohorts. Our best cohort, our best classifier rather, by machine learning sampled from all of these different features: clinical DNA methylation at 12 loci, 14 loci for copy number, and 63 transcripts. This was made by using 13 different machine learning algorithms, by cross-validation in the training set, looking at 23 million different classifiers. The best one had an area under the curve of about 0.8, and you can see how each of these different components, the copy number, methylation, clinical, and RNA combined. These are the individual ones in the colors. Black is combining all 3. That this classifier in the validation set now works whether you're sampling the low-grade area or the high-grade area of the cancer. And the next test for this classifier is to use it in biopsy material using how long the patient stays on active surveillance as our endpoint. I'm going to get back to that topic using other slides. So we were quite excited to learn about and get access to the new early access MethylSeek platform developed with Andrew Hatch and Lonnie Pickle as the leads at Thermo Fisher. So what this does is it takes 20 nanograms of DNA, pre-treats it with a well-controlled bisulfite conversion process that has some really nice quality control elements to it that we don't have and can't use for methylation-specific PCR. We had them make a 38 gene target panel for us with our genes of interest, including the pronto genes and those detection genes. We got 250,000 reads per sample and really good performance in terms of uniformity and mean depth. So this bisulfite treated DNA goes into something that looks a lot like the standard AmpliSeek process on the ion torrent with different bioinformatics. Andrew Hatch has a poster about this at this meeting. Thermo wants me to mention that this is for research use only, not for diagnostics. So let's compare the output of this panel to what we typically get with methylation-specific PCR. This is the RunX3 gene, which is part of the community methylation panel that's being offered by Thermo Fisher. And it just so happens that there's big differences between prostate cancer in red and benign prostate tissue in green. With methylation, we have to use normalized relative methylation. We don't have an absolute number, and that causes challenges from batch to batch. We also get these kind of wide error bars in the cancer samples. With the panel, we get percent methylation, which is quite easy, trivial to, it requires essentially no conversion from batch to batch. And that's because it gives you a read from the unmethylated strand and the methylated strand, or species. We get much tighter error bars, better separation, and we get single CPG resolution. So instead of an average across all these 9 CPGs, we get each one individually and can focus on our best one. We get the Watson and the Crick strand. We get separate information on both, so if one's better than the other, we'll be able to see that. So essentially we're getting something like 100 times more information and much higher quality information, not to mention the workflow. So we can only get 2 or 3 assays into a single tube with methylation-specific PCR. to work together, and they got 38 times 4, at least something, about 75 assays to work together in a single tube. So we're starting to explore uses of this technology in the non-invasive space. This is from sequencing with this panel. This is one patient across many loci looking at methylation levels in urine. And these are the 3 detection biomarkers I showed you, so we can see all 3, as well as some candidate prognostic markers from our Pronto classifier. We can also see RNAs in urine. So in conclusion, I think this platform is a major improvement for DNA methylation profiling, requires low input, has good uniformity, much better data, much better workflow. Also, we think adding methylation to RNA and copy number can add power to prognostic biomarkers and diagnostic ones, and this power should facilitate the development of better non-invasive tests. So I've thanked most of the people along the way. I want to thank our funders and the patients we study as well.